All right. So, what we're going to do today is sample macroinvertebrate communities, and we are at the Garrettsville wastewater treatment plant. So, the whole goal of the lab is to assess differences in macroinvertebrate communities between upstream areas, which is our control, and downstream areas of the water effluent, and I'll talk a little bit more about that in just a minute, to see what kind of impacts the wastewater treatment plant may have on macroinvertebrate communities. So, macroinvertebrate communities are really good indicators of water quality, as you may or may not be aware of, and you'll probably read a little bit more about it. Um, but we can use data from macroinvertebrates to tell us how we're scoring in a water quality aspect. So I mentioned in lecture that we were going to talk, or we were going to do a scientific paper this semester. And one thing that I want to mention is that this is going to go through multiple drafts. You're not just going to write it and it's done. It's going to go through peer review, it's going to go through edits for me. And what I want to focus on for next week is not only identifying the macroinvertebrates that we collect today, so we're going to bring them back to the lab with us. We'll be in lab next week. We'll social distance. I'll set up stations so that everyone's far apart. I have face masks. And again, talk to me if you feel really uncomfortable about that. I can set you up with your own situation to do your ID. So I don't want to talk too much about the data analysis today because we'll get into that more. But for next week, you are going to turn in a method section that is a preliminary method section. And it's going to cover what we're doing today. So it'll include a field site description and the sampling techniques that you're using today to collect macroinvertebrates. So what I have for you is in this pink bin, they're all sanitized, lots of clipboards. There's also some extra pencils in there if you don't have a pencil with you, and some write in the rain paper so that it can get wet and you can still write on it, that you can take notes on, because I'm going to talk about a few things methodology wise, and as it depends on your experience and your age and how your brain works, but I know that I forget things very easily, especially when I try to go back and write out a really full description. So it'd be good to take notes today, get that full description down on paper. You'll be able to take these notes with you. It's not going to be graded, it's just something that you get to keep with you. But you'll want to pay attention to site characteristics, and I'll talk more about that when we get down there. But let's go ahead and grab your clipboard. At the Garrettsville wastewater treatment plant. And what I'm going to do is tell you a little bit about the plant and their processing, just so you have an idea of kind of the system that we're dealing with. So up this way, upstream, there are containment tanks. This is an old abandoned one that was part of their old system, but essentially they first funnel their sewage through an anaerobic system. So there are a lot of anaerobic microbes that start to break down the sewage that comes in. And this comes in from residential areas in Garrettsville, but also businesses as well. So once it gets into those aeration systems, it spends some time there with settling and skimming, and then they have a lot of really cool giant carbon filters that filter the water. And at certain times of year, especially in the summer, before it comes back out into the river, it goes through several settling tanks. It's a whole setup. But sometimes they, and especially in the summer, pass it through UV light so that it can kill microbes. They're only required to do that when the count is excessively high. So they're under a lot of EPA regulations to self-report what their water quality is like coming out of their wastewater treatment plant. And that doesn't mean that they don't get spot checked here and there, but they have to do self-reporting, I believe it's every day or maybe twice a day, of their water quality. So as we get into winter, the river can handle different bacterial loads than it can in the summer, based on stream chemistry. So one thing that I do want to mention is that as you're getting ready to write your scientific paper for this class, you don't want to write it as a lab report that is so specific to this place. You want to focus your introduction very broadly on impacts of wastewater treatment plants on macroinvertebrate communities. And as you're thinking a little bit more about this place and the specifics of this place, that information is usually more relevant in the method section. So just to kind of keep that in mind. So what I want to do is walk you over to where the water is coming out of the So this is what we call our effluent pipe, E-F-F-L-U-E-N-T, effluent. So the wastewater that is treated comes through and gets discharged back into Eagle Creek. So one really important thing to understand is that, and we got to witness this morning, which was really cool. Their staff came down and took the water sample. They took it back to do fecal coliform testing, pH, dissolved oxygen, ammonia. They test it for a lot of parameters. And then they report that information to the EPA so that they're in compliance. If they are outside of compliance, typically what happens is that they get fined. And they had a really big issue a few years back where one of the industries in Garrettsville dumped a bunch of copper down their drains, and it contaminated the whole system here and it was something around $1 million plus tax dollars to essentially fix that problem. So, the wastewater was toxic, they couldn't release it, they had to settle it in bins and then find a place to dispose of that hazardous waste. And they do a really great job of making sure that water quality is up to standards here. So you shouldn't be afraid walking around in the river. There is going to be fecal coliform. There is fecal coliform literally in almost every aquatic system on the planet. And that is just a function of animals living in ecosystems. So I'll encourage you, obviously, to wash your hands and not, drink the water. Don't put your hands in your mouth. Yes, question. Coliform is a type of bacteria, a very specific type of group of bacteria that you find in digestive systems that pass through. So that's a reality. Almost every system is loaded with it, regardless of what you do. So one thing to keep in mind is that this morning we sampled downstream of the effluent, which is our treatment site. We only have 5 people in the morning lab, so we do need to collect a few more samples so that we can even out our downstream area versus what we're going to consider to be our control site that is upstream of the effluent. So we'll kind of break up into groups and figure out who's going to sample where. And the way that this is going to work is that we're going to stretch a meter tape and randomize the way that we're sampling. So I want to stop there and see if there are questions. Out a meter tape, and we're going to stretch it out 50 meters, and that'll be along the length of the river. So we'll do that both upstream and downstream since we're going to sample both areas. I have 2 different measuring tapes. And what we're going to do is envision a 1 meter square grid over the top of the stream so that we can randomly sample in areas. Remember, randomization is really important to eliminate bias. So what I have is a random numbers table. And I have 2 of them so that we can speed up the process a little bit. And what you'll see on each of them is that there are random numbers for length. So that will be the stretch of the stream in length. And then there's also a random number for width, which will cut across the stream in this direction. So, for example, if you get a length measurement of 36, you'll find 36 meters on the measuring tape in length, and then say your width is 3. We're gonna go facing upstream, which is this way, right side of the bank, and then you're gonna take 3 meters of steps to that location, and that will be your sampling location. Does that make sense to everyone? These aquatic D-net samplers, and they're called D-nets because they are D-shaped. Great. And what you're going to do is set them down in the stream so that water's flowing into the big part of the net. There's a big part of the net. There's a really small part of the net, non-sampling area. You don't want to try to collect things in this stupid small side. Not smart, right? You want to collect things in the big part. So what you'll do is make sure water flow is going into the big part of the net, and then you're going to kick in front of it, kicking up sediment, hopefully flushing in macroinvertebrates. You can shuffle water into it. You're going to kick around for exactly 20 seconds. So we have a standard protocol of 20 seconds. I do have some stopwatches. I have a handful of stopwatches, but feel free to, I'm not going to tell you to take your phone in the stream, but you can stand bankside with your stopwatch on your phone with a partner and shout out start and stop at 20 seconds. The sediment off. And then you're going to want to check your net to see if you have any invertebrates sticking on it. Okay? The reason that the water is important in here is that you'll see things moving around, and it'll be a little bit easier to see what you're trying to collect. So you'll want to live sort through your pan and collect everything that you can see. You may miss things. It's human error. That's just the way it goes. So there are some supplies here that will be useful to you as you're sorting through. And these are the viral wipes that I was talking about. We'll take them down there with us. I only have 4 stopwatches that are still working. They get wet and then they break really fast. I'll bring this. This is our trash bag to put viral wipes in. Some of the supplies that you will definitely need is a vial. And what I'm going to have you do is put your name and PM lab on the outside of your vial. There are Sharpies here that you can use. You're going to want to fill up your vial with a little bit of ethanol. This is 95% ethanol. That's probably an important value to write down. This is our kill agent. So maybe I shouldn't say this on video, but I'm going to say it anyways. Your invertebrates are going to have a super drunken day on this ethanol and get alcohol poisoning and have the party of their life. And then it's over. So, you'll want to fill up your vial with a little bit of ethanol. I would say, probably up to the bottom of this white square is enough. If you need a second vial, feel free to get it. That's going to be just fine. So you'll mark the outside with Sharpie. What I also have are some... Someone have a question? I heard some. Okay. I also have some tags for inside of the vials. So I always double mark my samples. So what can happen is that when ethanol gets on Sharpie, it runs. But what can also happen, so this is right in the rain paper, you want to use pencil on this. Put your name and your PM lab on it. But sometimes the interior labels get abrasion. And what I have found, and rubs off what you've written. So what I found is if I double up my efforts, one or the two always seems to work out just fine. So there are labels there to put inside of your vials. To collect invertebrates off your nets and out of your trays, There are forceps. Please keep the forceps in your inside pocket if you're not using them. What happens is that people set them down on the ground, someone steps on them, they get buried in the sand, and forceps are decently cheap, but I don't like a lot of eco waste. It's not good for us to do ecology and then leave, things in the field. So there are forceps here. Sometimes people like to use pipettes or spoons to help capture, so take one of each and see what you like. And then what was the other thing I was going to say about that? I think that's it with the sampling. So again, if you need more than one vial, that's just fine. Just make sure you label it correctly. You are going to take a total of 4 samples, and you're going to combine those 4 samples together. We're not going to keep separate tubes. And I have a lot of reasons for that I can share. But for now, we're going to keep everything in one tube.
Are you sampling the same spot 4 times?
You are not sampling the same spot 4 times. So you're going to pick 4 random locations. And again, I have 2 different random numbers tabled here. You're going to want to pick a spot that isn't already crossed off. So cross it off after you pick it. You're going to choose 4 lengths, 4 widths in order to sample. I would write those down just on your papers up here, and then you don't have to worry about it. And we'll start there.
Pipe while we're in the riverbed. Water's flowing downstream this way. Downstream. You see we have our measuring tape set up 50 meters long, hugging the bank of the stream. You see the end of it all the way down there. This is our downstream site. So I'll walk and show a little start. Notice it's relatively slow moving with some riffles up ahead, further downstream.
Oh, I thought it was 48 hours. But we could do it like that. That one.
The edge of the banks.
Yeah. Well, in that case, I would want to go all the way out. Yeah.
Riparian cover. Our effluent pipe again. I'm going to show you our upstream site. So here's this little sand bank with people going through looking for their macroinvertebrates. Hard at work, Grant. Good job. We have the beginning of our measuring tape here, and it goes further upriver. So you see the beginning of our tape measure is here, and comes through the beginning of a little riffle here. You see further upstream, it's slower moving. Our tape measure is just over that pipe there. Here's the side of the bank. Here's the other side of the bank for reference in writing your methodology, your field site description, riparian cover. This is our upstream. We show you the kick sampling technique and in the collection. Another good thing to point out is Eagle Creek is a 4th order river system. Something you will want to look into when writing your paper. And also the river, at least in our upstream site here and downstream site, is not very deep, though it is quite wide. It doesn't get more than a couple feet deep, as you can see. Still very much out of the water. Hey everybody, we have Grant here. He's gonna demonstrate the kick sampling method. So we're roughly 15 meters down the upstream site, and how many meters in?
10 meters.
10 meters in. Alrighty, I'm setting a timer for 20 seconds, and you're ready to start. 3, 2, 1. JC has the net facing upstream to where whatever he's kicking up there flows down into the net. We're obviously hoping to catch macroinvertebrates, but we do have the chance of catching crayfish and fish. Time.
Okay.
There we go. Here we see Professor Clark sifting through some substrate. Oh, looking for the macroinvertebrates.
There's one.